At Novavax, we are relentlessly committed to one goal: providing protection against the world's most urgent infectious disease threats. Protection for families, for communities, for the world. We are working to create best-in-class vaccines by focusing on what matters most: proving safety, establishing efficacy, trusted science, persistent hope. A protein-based established formulation is our foundation. An innovative recombinant nanoparticle technology platform is our difference. Developing adjuvanted vaccines against emerging and evolving infectious diseases is our strength. Our team of experts is working in partnership with leading organizations from around the globe to resolutely advance our pipeline so that whenever a vaccine is needed by anyone, Novavax is there. This is our commitment. Novavax.
This next session is a panel discussion on disruptive technology innovation and vaccine development. Please welcome your moderator, Kazem Kazimpur.
Wow. The lights are bright. Yeah. Okay. Good morning. I know it's a good time to be here after about a year and a half. My name is Kazem Kazimpur, just in case if you are looking on your book and say you are looking for Martin Rosendahl, I'm not. And I am President and CEO of Amarex Clinical Research, a local contract research organization that we have been heavily involved in COVID. Actually, the very first that we got approved was diagnostic, was in May 2020. And I'm very happy to be here and pleased to, an honor to talk to distinguished presenter that we're going to give you some insight in the vaccine development. Obviously, we know that really being in this COVID-19 helped a lot of us, those who are in clinical trial for the last many years, really will learn how to move as fast as we could. Not only in the design of the protocol, and as you heard very nice talk, previous presenter talking about all different things, doing it as much as we can in parallel, taking quote unquote risk except for the safety and efficacy of the product. And really, that has been in all cases. We, the clinical trialists, have done that, and regulatory helping us to do that, and that has really helped us to move as fast as we have. With having three great panelists here that they gonna give us some insight of how they could do it. I stop here and I ask my first, Bill, please introduce yourself and tell us a little bit about your background, your company, and how and what you have done through this process.
Sure, thank you. Thanks for having me. I'm Bill Enright. I'm the CEO of Vaccitech. Vaccitech is an Oxford, UK-based company. We're a clinical stage immunotherapeutics company, so mainly focused on the therapeutic side of things. We've got 6 programs right now in the clinic, or will be shortly, including a proof of concept study ongoing for a potential functional cure of hepatitis B, for a clearance of human papillomavirus, as well as a couple of oncology studies. But the program that kind of got us started on the COVID route was in a prophylactic vaccine against MERS. So the previous speaker mentioned some CEPI funding that went to Sarah Gilbert's lab at the University of Oxford. Dame Gilbert is our scientific co-founder, and so working with her on the development of that. So we were one of the only companies that had human data against a coronavirus vaccine. And so that really enabled us to move very, very quickly when the sequence came out for COVID-19. We were involved in the invention of this vaccine and then eventually out-licensed that to AstraZeneca through the University of Oxford. So that's how we play into it from the pandemic side. I've been in the Maryland biotech ecosystem for over 30 years. We were able to actually take Vaccitech public based on some of the results from the COVID-19 vaccine. So we raised $280 million this year and took the company public in April. That's my second public company. So I took Altimmune, which is a local Maryland company, public in May of 2017 as well.
Thanks, Bill. How about if I go to Mary Moran? She is joining us through Zoom. Mary, can you please tell us a little bit about yourself and your company and what you are doing?
I'm happy to. Yes, I'm happy to, and I'm delighted to be here today. Thank you for having me. I'm from Pfizer. At Pfizer, I am the North American Medical and Scientific Affairs lead for mRNA vaccines, specifically COVID-19 right now. Pfizer, as you may know, has a very long history of over 195 years of creating breakthroughs for patients. We have a very patient-centric philosophy and mentality. With regard to COVID-19, Pfizer partnered with BioNTech. BioNTech has proprietary mRNA technology. They are leaders in the field, and we used our expertise, our global network, our expertise in large-scale manufacturing, as well as our regulatory expertise, and work together to make the Pfizer-BioNTech COVID-19 vaccine, now licensed as Comirnaty. And what helped us was really that we had a partnership with BioNTech since 2018, at which time we were working on an mRNA influenza vaccine. And when the pandemic hit in 2019, we were able to take those 2 years of lessons learned and quickly apply them to SARS-CoV-2. And so that partnership has been a very good one and one that I hope to tell you a little bit more about later today.
Thank you very much, Mary. And our third panelist here is Robert. Can you please tell us a little bit about yourself, your company, and I know you're a data scientist and you can tell us a lot about data.
Yeah, for sure. Hello, everyone. My name is Robert Vandenberg. I'm head of data sciences and computational vaccinology for GSK Vaccines. We're located here down the road in Rockville. I've been with the company for since 2010, and I moved here in 2016. I've been working throughout my career on various vaccine platforms within GSK, so both adjuvants, chimp adenoviruses, and also on RNA. I work for GSK, so we're also a company with a long history, and also a long history of innovation within the vaccines world. I think we've licensed one of the first recombinant RNA vaccines, and we've been investing in new technologies all along. One of the most recent highlights is our Shingrix vaccines, which protects people against shingles. And we've been also active in the COVID response. We've been doing a dual strategy. So the first strategy was to offer our adjuvant, one of our adjuvants, to various partners that are developing protein-based vaccines. And we currently have 3 partnerships in the clinic. One is with Sanofi, a French vaccine company. One is with Medicargo, a Canadian company that is making plant-based vaccines. And the other one is with SK Biosciences in South Korea. And the second part of our strategy is also partnering with CureVac, which is also a European-based RNA company.
Thank you very much. I'm going to start asking some general questions that I'm sure many of our audience have that in mind. Do you like to go that approach, or you want to tell us more about a bit more insight? Which way do you like to go? Asking question and answer, or you tell us about the insight? And Mary, you too. how do you like we go through asking some questions that we all have that in mind, and I'm sure previous presenter answered some of those questions, but still there are some that we would like to hear from those who have hands-on.
I'm happy to be flexible. I did prepare a few slides just to give the audience an overview of where we've been and where we're going, but as I said, I'm flexible.
That's good. How about Mary? We go first with you giving us some slides, and then probably some of the questions that I'm going to have, I can raise them because you're going to answer them that way. So let's start with your slides, Mary.
That would be great. So if someone could put up my presentation, please.
Yes, it is on.
Terrific. So as I said, I'm very happy to be here to tell you about the Pfizer-BioNTech vaccine, also known as Comirnaty. Next slide, please. I need to begin by telling you our indication. The Comirnaty is a vaccine indicated for the active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 16 years of age and older. It's also authorized, so it's one vaccine with two names. Comirnaty and the Pfizer-BioNTech vaccine is authorized for emergency use to provide a 2-dose primary series in individuals 12 years of age and older, and a third primary dose, series dose in individuals 12 and older who have certain types of immunocompromise. A single booster dose is also authorized in individuals 65 years of age and older, 18 through 64 years of age at high risk of severe COVID disease, and 18 through 64 years whose frequent institutional or occupational exposure puts them at high risk of serious complications, including severe COVID-19. The two vaccines, it's one vaccine, same formulation, and can be used interchangeably. Next slide, please. So a little bit about our clinical trial. You may know that it is a global clinical trial. It's ongoing. Over 46,000 participants have been enrolled at 153 clinical trial sites around the world. You can see the age breakdown of the participants in the trial, remembering that we started with older individuals and then worked our way down to younger. And I'll tell you a little bit about our pediatric trial in those under 12 in just a bit. In addition to the United States, Brazil, Argentina, Germany, Turkey, and South Africa served as clinical trial sites. We did our best to try to recruit a diverse study population. Approximately 42% overall, that is globally, and 30% of U.S. participants have diverse, that is non-white backgrounds. Next slide, please. I selected some milestones just to sort of tell you about the journey that we've been on. Remember that in December of 2020, and I'll direct your eyes to the bottom of the slide. In December of 2020, the FDA provided emergency use authorization for persons 16 years of age and older. In May, the FDA expanded that authorization to include adolescents 12 to 15 years of age. In August, the FDA authorized a third vaccine dose for individuals with certain immunocompromising conditions. And on August 23rd, the FDA approved, that is, full licensure of the vaccine in individuals 16 years of age and older. And then just last month, the FDA authorized a single booster dose at least 6 months after completion of the primary series in individuals 65 years and older and adults at high risk of severe COVID-19. Next slide, please. Our vaccination schedule, the vaccine is administered intramuscularly as a primary series of 2 doses, 3 weeks apart in individuals 12 years of age and older, and a third primary dose, as I said, at least 28 days following the second dose in individuals at least 12 years of age who have undergone solid organ transplant or are diagnosed with conditions that are considered to have an equivalent level of immunocompromise. The booster dose, and this is what's on our label, which is different than what the CDC, somewhat different than what the CDC has recommended. The booster dose is provided as a single vaccine booster dose. It can be administered intramuscularly at least 6 months after completing the primary series to individuals 65 years of age and older, those who are 18 through 64 years of age and at high risk of severe COVID-19, and those who are 18 through 64 years of age whose frequent institutional or occupational exposure to SARS-CoV-2 puts them at high risk of serious complications of COVID-19, including severe COVID-19. Next slide, please. What the CDC interim clinical considerations say are the following. They really break down the booster recommendations into groups that should receive it. That is, it's a routine immunization recommendation versus ones who may receive it. That is, weighing the benefits and risks of receiving the booster based on an individual's condition. So those who should receive it, again, are people aged 65 years and older, residents who are aged 18 and older in long-term care settings, and people aged 50 to 64 with underlying medical conditions. Those who may receive it, based on their individual benefits and risks, are people who are aged 18 to 49 with underlying medical conditions, people aged 18 to 64 at increased risk of SARS-CoV-2 exposure and transmission based on their occupational or institutional setting. And that really is healthcare workers and other non-healthcare workers who are frontline essential people, essential for society. So teachers would be an example of that. And there's a full list of those who are defined in that group on the CDC website. Next slide, please. A little update about our phase 2/3 clinical trial in children 5 to 11 years of age. We've announced results from the trial in this age group using a 2-dose regimen of 10 micrograms, which is 1/3 of the dose of those 12 and older. 10 micrograms have been administered 21 days apart. And that 10 microgram dose was selected based on results of the phase 1 dose finding study that evaluated the safety, tolerability, and immunogenicity in that age group. We shared this data with the FDA for the agency's initial review. We will formally submit an emergency use authorization application very soon in the coming weeks. And we will, of course, submit data from the full phase 3 trial for peer review publication. Next slide, please. I wanted to just give a heads up. I'm not going to go in detail to this of this slide, but just to say that our current formulation, which is in the purple column, is one that is stored, as you may well know, at ultra low temperature conditions, can be stored for 9 months, 2 weeks in the freezer and 1 month in the refrigerator. We've been constantly working on new and better formulations that will allow the end user to use them more easily. We're still working in pandemic mode, so we're not at the pre-filled syringe point by any means. But our future formulation involves a different buffer called tris-sucrose buffer. That's the gray column. That's going to be a formulation that will be able to be used. It'll be stable in the refrigerator for 10 weeks. It does not require dilution. So it's basically thaw it and use it. And then we will have, people have asked, are we just going to use a smaller amount of the current formulation for pediatrics for if we do receive emergency use authorization, will we use the same preparation? And I just wanted to be clear that there is a new preparation, a new presentation of the vaccine that will be in an orange capped vial that will be a 10 microgram dose for younger children. Again, if we do receive emergency use authorization. Next slide, please. We've talked about, these are some of the things that we think about all the time. Just wanted to make the point that long-term safety will be monitored in our clinical trial participants for 2 years after the second dose of vaccine. As I said, we're working on new formulations and presentations to make it easier for the end user. Our booster dose, we'll be looking at it in terms of duration of protection, and certainly we've been looking at it for variant protection, and it is clear that at least in terms of levels of neutralizing antibodies that are produced by the vaccine with the booster dose, that they it's quite a robust immune response and pretty equal across variants, including the Delta variant. We have clinical trials. We talked about the study population of children under the age of 12 years. We also have a clinical trial for women who are pregnant and another for immunocompromised individuals over 2 years of age. We're looking at concomitant vaccine administration. We'll be looking at using the COVID-19 vaccine with other respiratory viruses, and so that's an exciting piece that we'll continue to explore. The messenger RNA platform, in my mind, is really a game changer. It offers an amazing degree of flexibility, such that we can be poised and ready if new variants of concern arise. And so it's a platform that once we know the genome, as one of my colleagues on the panel said, once we understand the the genome of a variant, we can quickly create a DNA template and quickly make the messenger RNA. So next slide, please. With that, I'll stop and happy to answer any questions, but I'll leave you with our important safety information. If you could advance the slide, great. And if we could just leave that on the screen for a bit. I'll turn it back to you.
Thank you, Mary. I appreciate it. Let me start with some question from you, Bill. What were the factors that enabled the rapid development of the COVID vaccine? We all know that when you start, you have to go through the process of writing the protocol, make sure that every component of it is there, sharing it with the FDA, waiting for their response, and then all that. And we all know that in no way we can cut corner with the respect to safety and efficacy. Both Mary said, the president said, the previous presenter said. What were the factors, what were the key factors that enabled you to develop it faster?
Yeah, I think as was stated previously, I mean, the experience that we had with coronaviruses in the MERS area in particular allowed us to have a good idea that that antigen, the full-length spike glycoprotein antigen, had a high potential for working. And so that really enabled us to, we didn't have to spend a lot of time looking for the right antigen and those kind of things. So, very similar to the RNA technology, ours is a viral vector-based technology. And we were able to move equally as rapidly, identify essentially as soon as the sequence came out, we ordered DNA and moved very quickly to make the viral vectors to get those into animal studies and start the important work in animals before moving into humans. And we were able to do that, extremely rapidly. So I think, we ended up in the clinic about 10 days after the RNA vaccines were in the clinic. And that's going through multiple animal studies, including non-human primates. So we actually waited for the non-human primate data before we started dosing people, because of some of the safety concerns around ADE at the time, and other issues. So, as you said, not trying to cut corners, but again, because we had government funding and other things, we were able to take the business risks to move things forward in parallel.
Business risk, not safety and efficacy. I like to emphasize on that part. How about you?
Yes, so we haven't gotten to licensure yet with GSK-supported vaccines. I think, so I will leave the others to comment on that. I think one of the things that really made a difference in pandemic responses is where everyone is really coming together, and that is what you were referring to in terms of regulators are stepping up. They really want to get to the vaccines, get into the market, and they really come and ask, okay, how can we help you make sure that we get all the right checks and balances in place, but move as fast as we can. I think also within companies, when you have such a crystallization point, resources start to focus and align on making sure that we can deliver on what is needed for. So there was, at least for our company, we had a very quick decision trying to make 1 billion doses of adjuvants available for partners that wanted to come and make use of it. And that type of decision making, that is really, that works when everyone is moving in the same directions, both from the funding agencies, from regulators, and also within the companies.
Thank you. Appreciate it. Mary, do you want to say, I know you already explained a lot, but can you tell us a little bit more?
Yeah, I would, first of all, I'm sitting here listening to my colleagues and thinking, gosh, how lucky we are that we have multiple vaccines to fight the pandemic. And so I got chills actually as I listened to people speak. I would just make two additional points to what's already been said. And that is that when we talk about the clinical trial and the seamless nature of it, what we mean is that we're doing steps in parallel rather than sequentially, which is the traditional paradigm. And by in parallel, I mean, and I'll just take our trial for an example, and I'm sure it's similar for other clinical trials. we were studying different doses in different age groups, in different intervals, all at the same time. So there, again, no cutting of corners, as my colleagues have said. The last piece is the large-scale manufacturing that happened with companies. Companies started to really ramp up production of the vaccine before they even knew that it worked. Right, for sure, right? Before we knew for sure that it was safe and effective. We had some preclinical data and we had early phase, 1 data, but that was the other piece that really got us moving. So thank you for that.
Thank you very much. I appreciate that. That parallel processing approach is the way to go. And actually, as we all know, when we submit in the traditional way, when we submit a protocol to FDA, we wait for 30 days to get a go-ahead or safe to proceed. But in many cases, we have heard from the agency within few days, and they reviewed it and gave us all the comments. With respect to what you mentioned, not doing the traditional of sequence approach and doing it parallel, while we submit the protocol to FDA, we kind of start finding the clinical sites, talk about manufacturing, although those, as you use the term, that business risk, that maybe the protocol is not valid. So, but we are taking all those steps in parallel, and I do really like that, and that's the way that we all did, and that's one of the main way that we could move things ahead without putting the safety and efficacy in any way or any danger. I can't help it, I'm going to ask these questions. These vaccines were developed so quickly. How can they possibly be safe? I know the answer, we are talking about it, but I'd like to hear it from all of you, because that's one of the things that I do, not too often, because we are here in Maryland and a lot of people here involved in clinical trial and all that. But still, I like to hear from all of you.
Mary, you probably have the most direct experience with that.
Yeah, I'm happy to talk about that. So, a couple things, and I understand why people ask, and we think about this in our own house. I have an 11-year-old, and so it's a great question. Firstly, for the clinical trial, as Kazem said, the FDA has been, and regulatory bodies around the world have been, a phone call away. I mean, we generally have been, what used to take weeks to sort of digest, it has been real-time safety monitoring during the clinical trials. We also have an unbelievably robust safety monitoring system in the post-authorization, or what typically would be called post-marketing period. The United States, and I'll speak specifically about the United States, had prior to the pandemic had about 6 existing safety monitoring systems, some of which were passive reporting. Others were sort of looking at large databases, Medicaid and such. And with the pandemic, they expanded, the government expanded safety monitoring systems, looking closly at long-term care facility data and other claims data. So, we're hearing about adverse reactions, and those are troubling to hear about, right? Myocarditis, anaphylaxis, clotting problems, but it's a sign of the tremendous safety monitoring that is existing. So we are finding out these things quicker than we would possibly otherwise. So I'll leave it at that and let my colleagues expand on that.
Yeah, I mean, I think the key thing is, as you noted in the numbers on your slide there, it's in tens of thousands of people during the clinical trials. So no corners are being cut in phase 1, phase 2, phase 3. Although the regulatory agencies did work with us in these adaptive trial designs, so that you were moving from one to the other much more quickly than in typical vaccine or even therapeutic development.
Thanks.
Yeah, but I fully agree.
Kazem, I was just going to add one last thing. The other thing that these clinical trials have had is an independent data monitoring committee. That is outside experts that have nothing to do, no financial relationship with the manufacturers, who have been monitoring clinical trial data continuously. So that's the other piece that I think it's important for people to understand. Sorry to interrupt.
Thank you.
No, I would like to take the point and build on it, and also in relation to the previous talk, because I think that the data collection for this pandemic was indeed unprecedented, and I really loved that I would get a text message on my phone to give my safety data as a real-life monitoring, which is great. I think when you start thinking about preparedness for the next pandemic, I think you also then want to start building on how you can generalize the findings, the databases from these trials and these vaccines to the next potential outbreaks. And I think for that one, GSK has been doing a lot of work in the past and still on understanding the biological mechanisms by which the vaccines work, and also to see if you have one vaccine against one target, how generalizable it is if you expand it to other vaccine targets. And I think that's going to be really important as well for the next one, for pathogen X, basically.
Thank you. I can't help but to emphasize some of the element that you mentioned and Bill mentioned, Mary, you mentioned, is that even in the slide you had phase 2/phase 3, and that is the quote-unquote seamless moving is not typical approach that we do. Phase 1, we finish the trial, read the data, analyze the data, and then take it to the agency, and then start writing the protocol for phase 2, and then finish phase 2, and then go to phase 3. The way that has happened is that, as you mentioned, doing data safety monitoring committee or doing interim analyses and presenting the interim analyses. Again, all this is because the safety and efficacy cannot be cut corner. So we use that interim analysis data, presented to data safety monitoring committee, who are absolutely independent from the decision makers in terms of that. And that really was one, and you also used the word adaptive clinical trial design. Again, that was another component that really helped to move from one phase to next phase, rather than taking that long time to do it. You do it as fast as we can. That really has been the case. And now, another question that I have is a bit different. I say that, are there other novel vaccine technologies in development or on horizon? I know that's a little bit, I'm departing from the current COVID, and we have heard all about it that other presenter mentioned earlier. Is there anything that you have in mind, and you can elaborate on that? I start with you, Bill.
So, we're obviously continuing to monitor those things. I think some of the interesting things you see now is kind of the hybrid approaches. Obviously, for Vaccitech, that's what we were working on the therapeutic side. We're big proponents of looking at heterologous priming and boosting, and I think you get better responses in both the antibody and on the T cell side. Overall immune responses with that approach, and so we're pretty much in favor of that approach, and I think we'll continue to see these new developments. I mean, again, one of the beauties of this stuff is that all the things that we're talking about that moved early and fast are platform technologies. I mean, they provide a lot of flexibility to be able to look at lots of different antigens very rapidly. And moving, that allows you to move into the clinic very rapidly. So when you're talking about that aggressive plan of a 100-day plan in order to get something to market, I mean, you couldn't even fathom that, 2 years ago.
Thanks.
Yeah, I love this question, and I agree that building on heterologous prime boost technologies is going to be, strategies is going to be really interesting in terms of presenting the immune system with different versions of the same pathogen, and possibly expanding the breadth of the immune response and convey a wider protection. I think other areas that we see a lot of activity happening is in, so now we deliver basically often the single spike protein. There have been efforts to make the spike protein more concentrated by formulating them in nanoparticles or in viral particles. And I think such a more concentrated presentation of the antigen to the immune system can also drive a stronger immune response, and possibly a more long-lasting one. And I think also, if you think about the fastness at which you can produce things, the next shift is really how can you screen multiple versions of proteins faster, and do more combinatorial experiments in your development.
Thank you. All right.
I have nothing to add. I think that my colleagues hit the nail on the head, and it's an exciting time. I think the only other thing we'll be looking at, or everyone will be looking at, I think, is the interval, right, between first and second doses for those vaccines that have 2 doses, and is there something that is a longer interval better for a more robust immune response?
Thanks. Just moving away from vaccine for a little bit, you mentioned about therapeutics. One of the things that we do see now, and it's becoming more type of question, is quote unquote the long haulers, or those who are after the. Are you using any of this methodology that we learned during vaccine for the treatment of the long haulers? And you know what I'm talking about, the long haulers. There are different terminology for the long haulers, and you may want to define what long hauler is, and then if you are using, any one of you, using any of the technology learned, being the Data Safety Monitoring Committee, being seamless, moving from phase 1 to phase 2 to phase 3, for the treatment of the long haulers. Obviously, there are so many symptoms and all that thing that from the long haulers. Any one of you, Mary, do you want to?
Sure, I'll take a stab. So long COVID is a continuation of symptoms or development of new symptoms for greater than 120 days from initial infection. And the symptoms you've probably heard or read about are things like, they can be a myriad of things, but include brain fog, fatigue, feeling listless. Almost every system can be involved, but there are about 20 symptoms that the CDC lists that are most common. Psychiatric problems, anxiety, depression, and such. We at Pfizer, and I'm the vaccine lady, right? So I don't know a whole lot about it, but we have an antiviral in the works that we'll be working on, and we're very interested in long COVID. And firstly, we're asking ourselves, if we prevent, certainly if we prevent disease, do we prevent long COVID? But if somebody ends up being immunized and then does have breakthrough, is there an effect on long COVID? That is, are they less likely to have symptoms of long COVID because they had been vaccinated and then had breakthrough infection? So we'll be looking at that, and we'll be working in concert with our antiviral colleagues to kind of begin to examine that.
Thanks, Mary.
Yeah, I mean, Vaccitech isn't working in that space in the COVID realm, but clearly, I think Merck presented some interesting data in the last week on antivirals, and other companies as well that are looking at the antiviral approach for dealing with long COVID and as alternatives to vaccination. Or not alternatives, but I guess in complementation to vaccines, because I don't think it's an either-or scenario. You need both approaches.
Absolutely, nothing really replaced. That's after having the symptoms and after having COVID. And as has been mentioned, that getting vaccine is the best, and preventing it rather than trying to treat it. Anything about the longer using? What I meant is that what we have learned during this, that we was really fast moving ahead. Are you using any of those what we have learned for either the treatment, as you mentioned, and or like the one from Merck is for treatment, and or for the symptoms left after getting COVID, as Mary mentioned, the brain fog, or even losing hairs as another element of it. There are so many of them, as CDC has mentioned, so many of them, with brain fog being one of them, and some other psychological effect. Is there any of the methodology that we learned during this vaccine period will apply to you have in mind to apply for the future treatments?
Yeah, I mean, one of the things people need to remember, I think, is that we're very early in this process. We're still learning a ton. It's 18 months in, or a little more than that now. But we're still learning about the duration of immune response from the vaccines, and as we said, still looking at what the right dosing intervals are. And there's just there's just a ton that we you know, in a normal course where you're developing something over a 10-year period, that we didn't have that luxury if we were going to try and save lives. And I think ultimately, you know, that was the biggest factor for all the people involved in this vaccine development, was trying to get it done as quickly and efficiently as possible to try and save as many lives as possible.
Thank you.
I don't have anything to add to that.
Okay, I don't have any more question for you. Let's see. Mary, is there any question that I left out that you have in mind you want to mention? Obviously, again, we talked about the treatment, we talked about the key factors, we talked about the using quote unquote the seamless from phase 1 to phase 2 to phase 3. Yeah, go ahead.
I think you hit the critical pieces, Kazem. I was so glad that you brought up safety, because that's such an important piece. And I think understanding this seamless and adaptive clinical trials is important. So I think you've nailed it, really. You know, what Bill said about how fast things are moving, it can't be overstated, I think. You know, people will say, why is the Pfizer vaccine, you know, so difficult to handle in terms of ultra low temperature? And why is the interval 21 days? And, you know, so it really is because we're in a pandemic. We did what we could as fast as possible to touch as many people as possible to save as many lives. And a short dosing interval of 21 days, again, that was to, let's try to get people's immune response up, let's get it going and save as many people as we can. So, yeah, we wish things moved faster for sure, and that we were in even farther along. And sometimes I lose sight of what we really have accomplished in that regard. So congratulations to all the manufacturers and all the scientists who have done so much in this regard.
Thank you. If you don't have any question, I'm going to open the session to audience. If anyone has some question for us that we'd like to learn from you, because I know a lot of you are very, very great clinical trialists. I've talked with you, and you have comments to say, and we can learn from you, or we can learn from the great panelists that we have. If you have questions, please go to the podium. There are several of them around. Okay, yes.
Hi, I guess I can start one. My name is Anshika, I'm with the NIH. I was just wondering that now that the vaccines are in phase 4 and we are actually giving it to many more people, are we observing any different adverse events than what we saw in the clinical trials?
Thank you very much for a great question. I'm sure you all heard that. The main question is post phase 3. Now, do we observe any adverse events? And if we do, go ahead.
Yeah, I'm happy to take that. Yes, we have. So myocarditis. Myocarditis we found is happens with the mRNA vaccines. It generally affects males, younger males, under 40, more than females, and usually after the second dose within 7 days. The myocarditis itself tends to be mild and is generally self-limited or requires only conservative management, but it's real, and we did not observe that in the clinical trial, and I don't believe it was observed in any clinical trial, even outside of Pfizer. So that's one example. Anaphylaxis. We did not observe that in the clinical trial. Severe allergic reactions. Yes, this is something that came up in the post-authorization period once the vaccine was used on, millions of people. And so, again, that speaks to the very robust safety monitoring process that we have. And there will be, this is, the nature of vaccines. There will be adverse events that we will continue to find now that vaccines are being given to a large amount. I will say that both of those events, anaphylaxis and myocarditis, are extremely rare. They're extremely rare, and we're seeing it because the vaccine has been given to, billions of people now. I think 6.3 billion people have been vaccinated globally. And so we're seeing it, it seems like we're seeing it quickly, but it's because so many people got vaccinated so quickly, right? So,
and the safety
great
monitoring
question.
is so good.
Thank you.
Yep.
Thank you. Any other questions?
Yeah, one more. I'm actually wondering about what vaccine companies are doing in terms of predicting variants, one, and how is that affecting the development of the current vaccine?
Thank you very much. About the new variants, and any one of you? And
yeah, I can. Yeah, so it's being monitored continuously, basically what variants are emerging, and basically we have epidemiologists and structural biologists that are monitoring how that will affect any future vaccine candidate that you want to move forward.
Yeah.
Yeah, I mean, it's the same. I mean, you're looking at, predictive algorithms and, trying to understand what are the potential variants that may come out, and trying to identify things that may or may not be protected by the current vaccines, and look at new vaccine formulations as a result, and then monitoring the situation as it exists.
I think to add one thing, so for flu we do this for years already. I think that COVID is clearly a different setting, but I think the tools and infrastructure that's been in place for flu can be also used for this type of monitoring as well.
Thank you. Mary?
Yes, we're watching, as my colleague stated, we're watching the variant situation very, very closely. The question is now, the Delta variant is the most prominent variant, right? It's over 95% in many states and over 90% across the country, across the United States. We know that the current version of our vaccine produces neutralizing antibodies that neutralize the Delta variant. So we're watching it closely, but are poised to be able to respond to new variants as they arrive. Had we learned that the current version of the vaccine did not produce neutralizing antibodies, did not provide protection against Delta for severe disease, we would be poised to create a new vaccine.
Thank you.
Hi. One quick question. We've been talking a little bit around how these technologies are accelerating the development of these things. I'm curious whether you think that's going to supplement or supplant sort of this approach as we think about other zoonotic diseases or other emerging pandemics, not necessarily coronavirus, right? Whether we think that we're going to try to reduce the timeline of development, or are we going to start looking at that sort of predictive piece? You talked about the variants, but predictive piece around things that aren't currently of interest, sicknesses for humans, I guess.
Thank you very much. Very nice question.
Yeah, I mean, I think it's actually a combination of multiple things, right? So you heard in the previous talk, there's work looking at trying to create vaccine banks, looking at a number of different coronaviruses or flus, other pathogens or zoonotic diseases that we think may be relevant, starting to create banks of vaccines so that we have a faster starting point. I think some of the things that we lessons learned in this will be applied in those other the next pandemic, because unfortunately it's not if, it's when the next pandemic will occur. And so we've got a lot of good lessons learned. I think what's interesting is how much of that will be applied to drug development in general, or vaccine development in general moving forward, and whether or not we can cut some of the costs and cut some of the times in our standard stuff, not pandemic related. So that I think is at least from the early signs, is going to be more problematic, where we thought we could utilize some of those things, with the regulatory agencies and clinical trial designs and things with other new drug and vaccine development. I think that's going to be harder than it is during a pandemic setting.
I think to build on that, because I really enjoyed the CEPI talk, and I think what is key is partnership there, because you want to have a portfolio of both disease targets, but also of technologies, because it's not guaranteed that each technology will work for each indication, and building these banks with safety data up to a certain point for a sort of a top list of potential pathogen access will be really useful to kickstart the next response. And I think there is an opportunity here as a global community to really partner up, because after the flu pandemic in 2010 and the Ebola pandemic, you see a lot of enthusiasm and willingness and good effort to go and build these coalitions. And then typically it tends to peter out a little bit, the incentives. And I think that with this pandemic, I hope we can really take good strides in the direction of building that infrastructure and those capabilities.
Thank you very much. If there is no other question, it seems that there is one.
I have a question. So I've seen that a lot of people are mixing vaccines and they're administering different vaccines. So I'm curious to see what is the clinical data and how effective it is when people, like, say, administer Pfizer and Moderna or like mixed vaccines.
That's a very good question. I don't know if any one of you is ready talking about the mix. I know an FDA advisory committee going to talk about it in next few weeks. But
14th and 15th, I think.
14th and 15th, yes, I know that. But yes, any one of you wants to address that very, very nice question. Thank you.
It's a great question. I was just going to say that the NIAID will be putting forth data on mixing and matching the currently authorized vaccines very, very soon. Probably, as you said, in the coming week or 2 weeks, 3 weeks.
Yeah, I mean, again, you know, as I mentioned, Vaccitech has been involved in that heterologous prime boost for a long time. This is something that's been studied for, you know, more than a decade, looking at various heterologous prime boosts, not in COVID-19, but you know, from a therapeutic approach, also from a prophylactic approach. So I think there's a lot of good science that already exists showing that you get improved and robust immune responses using that approach. And you know, but as with everything, it's got to be specific to the indication here. So I think that those data are forthcoming.
Thank you very much. I appreciate everybody, and that was really, I learned a lot, and thank you very much, everybody. Have a good day. Thank you.
Thank you.
This year's Maryland Life Sciences Bioinnovation Conference is sponsored by Johnson & Johnson Innovation. J-Labs at Washington, D.C. is a life science and healthcare incubator located at the new Children's National Research and Innovation Campus. J-Labs at Washington, D.C. serves as the hub for Blue Night, a joint initiative between J-Labs and the Biomedical Advanced Research and Development Authority. We'd like to thank all of our sponsors for their support of this year's Maryland Life Sciences Bioinnovation Conference.
Breakthroughs in science demand exceptional expertise and constant innovation in the pursuit of life-changing results. From discovery to delivery, scientists and research professionals worldwide trust Avantor to provide the mission-critical products, services, and solutions they need to move their research and discovery forward. This is our purpose. This is our mission.